Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about cell cycle checkpoints. Cell cycle control. What is cell cycle? We are going to discuss the role of cyclins and CDK inhibitors, PRP phosphorylation, regulation of cell cycle, and at the end we are going to discuss about checkpoints. Cell cycle is a precisely programmed series of events that enable the cell to duplicate its contents and to divide into two data cells. This series of events is controlled by the machinery that is often termed the cell cycle clock. So, if we talk about role of cyclins, specific steps of the cell cycle are controlled by changing the levels and availability of cyclins. Cyclins function by activating the catalytic function of their partners, the cyclin-dependent kinases. CDKs is a family of serine-threonine protein kinases, and on activation triggers the downstream targets. And if we talk about the CDK inhibitors, additional control of the cell cycle is provided by CDK inhibitors, which antagonize the activities of cyclin CDK complexes. CDK inhibitors are abbreviated as CDKI and are main player of the cell cycle control because these inhibitors are necessary for stopping the cell if its genetic makeup is altered or if its growth is not as appropriate for dividing into two daughter cells or completing this cell cycle. They apply brakes to the cell cycle. We are going to discuss them in detail in the later videos. And PRV phosphorylation protein retinoblastoma, that its phosphorylation also controls the passage through the R point. Hypophosphorylated PRB blocks passage through the R point restriction point, while the hyperphosphorylated PRB permits this passage. Means to say, if retinoblastoma protein is phosphorylated at higher extent, that will allow the cell to progress from the R point and to enter into the remainder of the cell cycle. And if retinoblastoma protein remains hypophosphorylated, means to say the phosphorylations are not at the appropriate or higher levels, the PRB will not allow the cell to enter into the further phases of the cell cycle. That will block its passage, that will block its way to continuing from the cell cycle after the R point. So that is our main point that we are going to discuss in today's video is about cell cycle checkpoints. This is part of the cell cycle control mechanism in all eukaryotic cells. Checkpoint controls operating throughout the cell cycle ensure that a new step in cell cycle progression is not undertaken before the preceding step is properly completed. Many types of cancer cells have inactivated one or more of these checkpoint controls, thus helping themselves to accumulate the mutant genes in altered karyotypes that propel their neoplastic growth. Neoplastic growth means to say that is their near growth, new cell formation. So there are three checkpoints that are important for the cell cycle control. Are G1 checkpoint that is located at the G1 to S transition between the R and S phase. So the G2 checkpoint at the G2 to M transition, the spindle checkpoint at the mitotic or phase transition. So there are the checkpoints as you can see in the diagram. G1 checkpoint checks for nutrients, growth factors, DNA damage, and if any of these is not normal, the cell will enter into GO or resting phase. And after that, you can see here after the G2 ending, before G2 ending, there is G2 checkpoint that checks for cell size and DNA replication. Because DNA replication occurs in the S phase, it is checked on before entering into the mitotic phase. And in the metaphase to checkpoint that checks for chromosome spindle attachment, that before metaphase, after that the next step is about the anaphase, that is going to split the chromosome. If chromosomes are not intact, that will also halt the cell proliferation or stop the cell cycle. So at G1 checkpoint, following things are checked by the cells. First of all, size. Is the cell large enough to be divided? Like its growth is at the appropriate levels? Has it gained the nutrients as well? Does the cell have sufficient nutrients to sustain the division? And it also checks for DNA integrity. Has the cell intact DNA or is damaged at any point? Molecular signals are also checked, such as cell receives positive signals from the surrounding to proceed for division. And D-type cyclins or extracellular signals. What is their role in the checkpoints? If we are going to discuss in detail, the levels of the D cyclins are controlled primarily through the extracellular signals. The remaining cyclins operate on a decided schedule. As we have seen in the previous videos about the cyclins, that what is their role and how they are changing from one stage to the other. In G1 phase, the D-type cyclins are prominent, and at the R point, the E-type cyclins are prominent, and after the R point, the A-type cyclins, and then after into the G2, G2 to the M transition, the E-type cyclins are the more important. B-type cyclins, sorry to say. B-type cyclins are prominently active throughout the remainder of the mitotic phase. So if a cell does not get move forward signal or go ahead signal, it shall leave the G1 stage and enter the GO or quiescent state. Deregulation of the restriction point decision making machinery accompanies the formation of the most types of cancer cells, since it leads to unconstrained or uncontrolled cell proliferation. So if we talk about the growth versus quiescence, decisions concerning growth versus quiescence are governed by the state of phosphorylation of three pocket proteins: protein retinoblastoma, protein 107, and protein 130. These proteins in turn are controlled by D cyclins and cyclin A. PRB, the retinoblastoma protein, controls passage through the R point. Hyperphosphorylated PRB blocks passage through the R point, while hyperphosphorylated PRB permits this passage. So PRB phosphorylation is carefully controlled. Growth factors induce expression of D-type cyclins, which initiate PRB phosphorylation by phosphorylating it. PRB acts by binding or releasing E to F transcription factors associated with promoters of genes that assist the cell from late G1 into S phase, helping them to cross the restriction point. Hyperphosphorylated PRB binds E2FS, while hyperphosphorylated PRB releases them. When viral oncoproteins are present, they mimic PRB. They act like PRB, but they are not allowing PRB to phosphorylate. So hyperphosphorylation by preventing PRB from binding with transcription factors such as E2FS. So if that was the first checkpoint, and now if we talk about the G2 checkpoint, this checkpoint ensures that before M phase, and it assesses the cell following events like integrity of the DNA, replication of DNA. If DNA of the cell is damaged, the checkpoint will arrest the cell and trigger the DNA repair machinery to repair the damage. If the damage is irreparable, then the apoptotic machinery of the cell is switched on to kill the cell, so that damaged DNA is not passed to the next generation, because that will lead to the cancerous cells. So moreover, DNA is also checked whether it is completely replicated. If not, replication machinery is brought in action. So that is the last checkpoint, that is metaphase checkpoint. This checkpoint occurs at metaphase, and it is regulated by APC. APC stands for anaphase promoting complex. APC is one of the ubiquitin ligases which are mainly required to induce progression and exit from the mitosis by inducing proteolysis of different cell cycle regulators. These cell cycle regulators or enzymes, or these all things that are controlling the cell cycle, are actually the proteins. Those their proteolysis is carried out by ubiquitin ligases. This checks whether all the chromosomes are attached to the spindle or not. It also checks the integrity of the spindle. If these events are not intact, then the cell will be halted until the cell repair is its defects, or if it does not repair its defects, it is arrested and cell enters into GO phase. So that was the video for today. We will meet in the next video. Till then, goodbye.